Hello dear students, let me start with the second lecture about general embryology. And let me start at this moment when previous lecture was finished. That means at the end of the second week. You can see that it is the 14th day of the development, so the last day of the second week. I'll briefly describe this stage, which you already know, and then I will go on through the third week, fourth week, and I will focus mainly to the structures which are developing not only during these 2 weeks but also later, but which are not included in another lectures. That means the development of muscles, bones, limbs, and also body cavities. So this is the program of the lecture, and let's look at the picture describing the situation at the end of the second week. The embryo at the end of the second week is in the stage of bilaminar germ disc. This is this structure here, bilaminar germ disc, which is composed of two layers, which you already know. This blue, higher, columnar, simple epithelium is a epiblast, and this yellow, low, cuboidal or flat, simple epithelium is called the hypoblast. This is the composition of the bilaminar germ disc. Bilaminar germ disc isn't alone here. It is surrounded by its two cavities. The cavity above the epiblast is called amniotic cavity. Amniotic cavity is here, this smaller cavity, and it is bordered by epiblast and by flat cells derived from embryoblast, developed during the process of dehiscency of embryoblast, which are called amnioblasts. The bigger cavity here down under the hypoblast is called a yolk sac. It was developing through the primary yolk sac towards the secondary yolk sac. Now there is a secondary yolk sac because there exists also exocelomic cavity, which was developing during the transformation of the primary yolk sac to the secondary yolk sac, and which contains some cells which were before this transformation part of the wall of the primary yolk sac. So it's possible to say that the young secondary yolk sac is a little bit smaller than was the primary yolk sac. All these structures, that means amniotic cavity and yolk sac, are surrounded by loose tissue, which is gray in this picture, not so well visible as in another schemas, but it is visible. This gray tissue, which is forming envelope of the whole embryo, but also connecting stalk, connecting the embryo proper with the extraembryonic layers, and also covering the inner side of another cavity which I will describe. All these cells here, all this tissue here is called extraembryonic mesoderm. Extraembryonic mesoderm. It isn't written in this slide, nevertheless you already know extraembryonic mesoderm. Extraembryonic mesoderm is developing during the second week. And the first source of the extraembryonic it isn't full. The first source of this mesoderm is hypoblast. Later, some epiblast cells will participate here, but now at this moment, the whole extraembryonic mesoderm derives from hypoblast. The extraembryonic mesoderm at the moment of its development was quite compact layer, but during the growth of mainly extraembryonic structures, trophoblast, trophoblastic lacunae, which are filled with blood, and so on. This compact extraembryonic mesoderm splits into two layers, and a space is developing between them, and this space becomes very big. The space between two layers of extraembryonic mesoderm is called extraembryonic coelom, or you maybe already know another name which is maybe more used, chorionic cavity. So extraembryonic coelom or chorionic cavity is a new cavity which is developing at the end of the second week and which is situated between two sublayers of the extraembryonic mesoderm. These two sublayers are called extraembryonic somatopleuric mesoderm or somatopleuric layer, which lines the trophoblast, which forms also connecting stalk, and which covers the amniotic sac, amniotic cavity, very small at this time. And the second layer is called extraembryonic splanchnic layer. I'm looking here, but I don't see this name, but it is this layer which is surrounding yolk sac. It is extraembryonic splanchnic mesoderm or splanchnoplura also. Splanchnic or splanchnopluric. All these terms can be used. And this is only around the yolk sac. It forms also this yolk stalk, which is temporary structure containing exocelomic cyst, but it quickly disappears. So after the disappearance of this stalk here, this so-called yolk stalk, the embryo is connecting with the chorion by only one connecting stalk. Before this event there were two stalks, one made from somatopleuric mesoderm, another one made from splanchnopluric mesoderm, but now there is just one after the disappearance of this stalk. So I think that it is the situation which you already saw at the end of the previous lecture, situation at the end of the second week, and we can go on. We can go on and we can start the third week of development. The third week of development is typical by three important events. Gastrulation, which is formation of three layers, so the embryo is changing from bilaminar to trilaminar germ disc. Then there is also notogenesis, process when notochord is forming and when axial structures are established. And the third very important process, which is beginning during the third week but is going on during the next week, is neurulation, and it is formation of nerve tissue, neural tube, and mainly central nervous system. So gastrulation, notogenesis, and neurulation are three important events which are which have place in the third week, or at least are starting as a neurulation in the third week. You can see that the length of the embryo is increasing during the third week approximately threefold. So at the end of the third week it has about 1.5 millimeters. But of course it's the size of only embryo proper, so of only the derivative of two main layers, nothing more. The whole conceptus is much bigger. Let's go to the 15th day, so the first day of the third week, and let's look at this dorsal surface of the bilaminar germ disc. This is still bilaminar germ disc, blue epiblast, yellow hypoblast, but something happens in the epiblast. new structure is here forming. It is so-called primitive streak. Primitive streak is a new structure. It is a thickening of the epiblast in the midline, which is situated in the future caudal part of the embryonic disc. So when this thickening, long thickening, sagittally situated appears, we recognize, yes, here will be caudal end. Near the primitive streak, later, let's say 1-2 days later, cloacal membrane will develop. It will be a structure, disc-like structure, where epiblast and hypoblast are connected by tight junctions, and something similar is developing near the cranial end of the embryonic disc, but it will be called oropharyngeal membrane. So cloacal and oropharyngeal membrane, but really it is well written here. These two things here are only future positions of these membranes, because we cannot find them during the 15th day. During the 15th day there is just beginning of the formation of primitive streak. Later, another structure is developing in the cranial end of the primitive streak. It is also thickening of the epiblast, but little wider and higher, and it will be called primitive node. So the second primitive thing will be primitive node. So then we'll have primitive streak and node at its cranial end. The cells which are proliferating are at first forming just thickening of the epiblast layer. Epiblast was, before the beginning of this process, simple columnar epithelium, and now we have got thickening, the place where we'll see in the section stratified epithelium. So that is, but the cells cannot increase in number still more and more, but if there is some threshold of this thickening, the cells become invaginated. I must use another color now. The cells become invaginated deeper. So these cells don't go up and up and up, but go down and invaginate, and the structure changes from the primitive streak into the primitive groove. So, or maybe better is to say that in the center of the primitive streak, a new thing, primitive groove, is forming. And the same is in the primitive node, where primitive pit is forming in the center. So let's imagine that at the beginning it is just thickening, and the second phase is formation of groove or pit. So something like this happens, and this groove or pit becomes still more and more deep, deeper and deeper. Okay, and the formation of primitive streak and groove is the beginning of gastrulation, and formation of primitive node and pit, it is the beginning of notogenesis. I explain it. So let's go to gastrulation. This is the situation at the end of the 15th day, let's say. Primitive streak with groove in its center. And what is here? Cells are invaginating and migrating, and they are migrating down, and they are migrating to the hypoblast layer. Here is hypoblast, layer of lower cuboidal epithelium, and the cells, new cells from epiblast, from the bottom of the primitive groove, are migrating to this hypoblast and transform this layer into new layer. The cells of hypoblast are interchanged by new cells developing from epiblast, and this new layer will be called endoderm. So now we have got a situation that there is still epiblast, but ventrally isn't already or isn't more any hypoblast, but a new endoderm. It happens at the very beginning of the gastrulation. Let's say 1 day later, endoderm is fully developed, and you can see that it is in fact morphologically the same as was the hypoblast. Again, one layer of cuboidal cells, but these cells are new. These cells are from epiblast, so it isn't the same layer, it only looks like it. And so we can say that it's definitive endoderm now. And the gastrulation is going on. This invagination and migration of cells doesn't stop at the end of the 15th day. So during the 16th day, these new cells are filling the space between epiblast and this new endoderm. This space is quite free, there aren't any borders, the epiblast and hypoblast are only very loosely connected, so this space is quickly created and new layer is forming in between, and it will be called mesoderm. And because we already have extra-embryonic mesoderm at this time, it was developing a week earlier, we can say that this new layer should be called rather intra-embryonic mesoderm. So let's add this word to this picture. Intra-embryonic, I can abbreviate it. Intra-embryonic mesoderm to distinguish it from the extra-embryonic one. Okay, so what have we got now? We have a disc which hasn't got 2 layers but 3 layers. Its layers are completely new layers are endoderm and intraembryonic mesoderm, and everything what is remaining now is to rename the epiblast. Epiblast is still the same, there isn't any interchange of cells, but now at this moment we can change the name of the epiblast, and the epiblast becomes ectoderm. So trilaminar germ disc was developed, composed of ectoderm, mesoderm and endoderm. I think that you already know these terms. It is taught in grammar school, or not, maybe not grammar school, but secondary or high schools. I think you know it. Ectoderm, mesoderm and endoderm, layers of trilaminar germ disc. And let's go on. The cells of mesoderms are migratory. They lose their contacts, they lose their cell adhesion molecules, and they migrate according to these arrows. They migrate to the whole space between ectoderm and endoderm. Now we can use these terms, and they fill it. They even migrate to the most cranial area of the embryonic disc, to this zone between a new oropharyngeal membrane and the rim of the disc. It will be later so-called cardiogenic area or cardiogenic zone, and you recognize from the name that the heart, sorry what I'm writing, cardiogenic area. You recognize hopefully that heart will develop here, but later. Let's let it for another lecture. So The mesoderm is filling the whole space between ectoderm and endoderm, with the exception of two locations, two membranes, where tight junctions are developing between endoderm and ectoderm, which they were developed already between epiblast and hypoblast, but then change of hypoblast are transformed or interchanged by new cells, so then there is a tight junction between ectoderm and endoderm in these two places. So mesoderm isn't here present. It is very important because these two places, oropharyngeal and cloacal membrane, these will be later connections of inner and outer space of the embryo. It will be cranial and caudal end of the digestive tube. It's logically logical from the names. And now, what happens in the area of primitive node? Primitive node at the cranial end of the primitive streak is also invaginating. Primitive pit is here forming, and this primitive pit becomes still deeper and deeper and deeper because the cells are still proliferating in the deepest parts of the pit, and these cells don't lose their cell adhesion molecules. Cells are still epithelial, and so it isn't possible to say that these cells migrate, but the cells rather increase in number, and the whole structure is growing, and this whole structure is growing only cranially in the direction of this arrow, in the cranial direction. And sorry, what's happened? So only in this cranial in this cranial direction the structure is growing, and this structure, this solid cord of cells which is growing towards the oropharyngeal membrane from the primitive node is future notochord, but now we can say that this is just pre-notochord. Pre-notochord, the cells forming the pre-notochord are pre-notochordal cells. So pre-notochord, or another name is also possible to find, and it is chordal mesodermal process. But okay, let's stay in pre-notochord. Okay. So this is also the beginning of notogenesis. But let's finish gastrulation. Cells are migrating and filling the space between ectoderm and endoderm. The whole embryonic disc is changing its shape. At the end of the second week and at the beginning of the at the beginning of the third week, it was circular, let's say. Before the formation of primitive streak, there wasn't possible to recognize the future left, right, cranial, caudal end. Now, after the formation of the streak, it is possible. We know that here will be caudal end, so logically here must be cranial end of the disc. Here is its right side, left side. But the whole structure is quite symmetrical, circular. But during this third week, the disc elongates and its cranial part becomes bigger and bigger. So it's changing its shape into pear-like shape. It is like a pear with a bigger part in the cranial side. Some histological slides from this very early development. It is a very young embryo. We can see epiblast here. This is epiblast. We can see thin hypoblast here. And this group of cells, it must be cells which are developing from primitive streak, which is here probably, and which are migrating to the hypoblast and changing it to the endoderm, or which are migrating to the space. We can see some cells here and forming very loose at this moment mesoderm. Then we have got several a little bit older stages. Here is a developing notochord, for example. Okay. During the third week, of course, many signal molecules are working because body axes are developing. Craniocaudal axis is given by a sequence of Hox genes, you know it, and these Hox genes as the second messengers stimulate the formation of another molecules. Some of them are transcription factors, some of them are signal molecules, some of them are expressing in the primitive node, which becomes center establishing the caudal end of the embryo. Another molecules and other factors are developing here in the cranial end of the embryo, in the area which is called, not very perfectly, anterior ventral endoderm, but it is called endoderm already, anterior visceral endoderm, but it is called visceral endoderm already when hypoblast is here, but it doesn't matter. So there are two centers of AVE, so anterior visceral endoderm, important for the formation of cranial end, and primitive node, which is important for the formation of caudal end of the embryo. Everything is modified by the gradient of BMP, which you know that it is you know from the molecular aspects of development that it is inhibitory molecule. So the higher level of BMP4 is, the lower level of these factors expressed in primitive node are, and this low level of these factors is important for so-called ventralization of mesoderm. On the other hand, high level of these molecules here are important for dorsalization of mesoderm, formation of notochord, paraxial mesoderm, which are developing mainly from the area of primitive node. So it is formation of the longitudinal axis, craniocaudal axis of the body. Quite interesting and very important is also formation of left-right asymmetry of the embryo. You know that the human body is asymmetric. For example, heart is more in the left side, liver is right, stomach is left, spleen is also left, appendix is at the right side usually. So the body is asymmetric, but regularly asymmetric. The same asymmetry in everybody. How is it established? It was recognized and discovered that a big role here is played by special signal molecules, which are expressed in the primitive node also. So the node is very important center for formation of the embryo. These factors are secreted to the surface of the node and transported to the left side by the movement of special cilia, which are situated on the surface of epithelial cells forming the node. These cilia are different than you know from our general histology, because there aren't two central microtubules. These microtubules are not here, there are only 9 peripheral doublets. Nevertheless, the cilia are movable, they are moving, their movement is circular, and the fast movement, component of the movement is usually from right to left side, and so the gradient of these factors secreted by cells of primitive node becomes higher and higher at the left side and lower at the right side. Each cell of the primitive node has got just one nodal cilium. This is the nodal cilium. You can see that it's quite short, and it is rotating. It is rotating when you open our Moodle page. You'll find in the topic of general embryology 2 very short movie, or it is only a link to this movie on YouTube, where you can see movement, circular movement of cilia, and from after you will see this movie, you can really recognize how the gradient of these factors is forming and increasing, and this is the principle of formation of left-right asymmetry. So cilia are important every times. Now we go to notogenesis, second important event of the third week. It starts little later than gastrulation. Gastrulation has started at the 15th day, notogenesis, let's say, on the 16th day, 1 day later, and the beginning of the notogenesis is deepening of the primitive pit. Primitive pit was at the beginning quite shallow, but it becomes deeper and deeper, and the structure forming here is called notochordal process or pre-notochord. At the beginning is a solid cord of cells, but lumen is developing inside. So in the section, cross section, it is then hollow structure. This lumen, this channel in the center of the notochordal process is called Lieberkind's channel. I write it. Lieberkind. Maybe you already heard the term Lieberkind crypt. It is called according to same scientist. Lieberkind crypts are in the intestinal mucosa, and Lieberkind canal or Lieberkind channel is embryonic structure developing at the beginning of notogenesis. The notochordal process grows cranially, but stops its growth near the oropharyngeal membrane. It is here, because here tight junctions are developed between ectoderm and endoderm, and so the growth more cranially is impossible. Before the notochordal process is reaching this area, another cells are migrating here, probably also from the primitive node. These cells are very important inductive cells, inducers. They are called Prejordal Mesoderm or Prejordal plate also. These cells are at the beginning situated in the mesoderm near the oropharyngeal membrane, but then they incorporate themselves into the endoderm of the oropharyngeal membrane. So you will see next picture, they are then in the endoderm of the oropharyngeal membrane, which becomes at this place little higher, columnar, and that's why this structure is called prejordal plate. Prejordal plate is something else than pre-notochordal cells or pre-notochordal process. It is group of cells which is serving as the inducer of the notochordal development, which are serving for hemotaxis of cells of the future notochord to grow cranially only and not to another side, and it is also very important for the later formation of nerve tissue and brain. So prechordal plate is very important, but not many things are known about its origin. So every textbook says something else, but probably they originate in the tissue of the primitive node. The next step, you can say, 1 day later, it was 70 day, 18th day now. During the 18th day, the next step of the formation of notochord is here. The ventral wall of the notochordal process is fusing with the endoderm, and both these tissues become interrupted after this fusion, and communication between yolk sac, which is here, and amniotic cavity, which is here, is forming. This communication, which is fully developed during the 19th day, is called Neuroenteric Channel, Neuroenteric Canal. Neuroenteric Canal is called because it is connecting the area of future nerve tissue and future enteros, that means gut. Gut is developing from the endoderm, from this roof of the yolk sac. Nerve tissue and brain and spinal cord is developing from the ectoderm here. So logically it is connecting future spinal cord and future intestine, future gut. But it's only very temporary structure. It develops during the 18th, 19th day, and it disappears during the next day. So till the end of the 3rd week it completely disappears, but it can sometimes remain and give rise to some developmental errors. After the formation of this neuroenteric channel, the notochordal process is connected with the endoderm and with the yolk sac cavity. So we can see, let's say, just half of the former full notochordal process, and this half process, this structure, is now called notochordal plate. So the first stage of notochordal development is called notochordal process, or also prenotochord. The second stage is notochordal plate. And during next days, 2 days, let's say, this communication disappears. Endoderm is healing, if it is possible to say, so becomes again continuous. And our cells here are forming again solid cord, and it will be now called definitive notochord. So notochordal process, first stage, notochordal plate, second stage, notochord, third stage. At the beginning was the solid cord, at the end is the solid cord, but in between is the hollow structure and plate, and so the development of the notochord is quite complicated and interesting. Picture from another textbook, but almost everything is the same. Only I want to emphasize this prechordal plate, which wasn't so well visible in the previous picture, really thickening of the endoderm in the area of oropharyngeal membrane. Before a while it was a group of cells here, but now these cells are incorporated into the endoderm in this area. And it is the situation of the notochordal plate and communication, that means neuroenteric canal. cross-section at the same moment, so notochordal plate we can see here, fusion of endoderm which is interrupted by this situation, and notochordal cells. And once more, completion of endoderm and fully developed notochord is in the center. Why notochord is so important? Why I am so long speaking about notochord? Notochord is very important because it is typical for all chordata, that means animals where human species is belonging. It is important axis. It is stimulating many other tissues for the formation of or for the differentiation. Maybe two structures are the most important which are stimulated by notochord, and that is the formation of neural tube and vertebral column. Without notochord, vertebral column and neural tube later transforming into the spinal cord wouldn't develop, and so that's why the existence of notochord is the reason why we got the spinal cord and vertebral column. So very important structure. But notochord in fact almost completely disappears at the end of the embryonic development. The only remnants of the notochord in adult are maybe, but probably as cells in the nucleus pulposus of intervertebral discs. Nothing more. But during development, this structure is very, very, very important. Picture which we already saw, but higher magnification only here is a notochordal process with a lumen. This lumen, this space here, it is a Lieberkind's canal, and one day later maybe, or two, fusion of the notochord and endoderm. So this is notochordal plate here. This structure is notochordal plate. You can see interruption of endoderm at this place. We'll go more again notochordal plate, but later neural tube is developing above, and here notochord situated again back in the central layer, that means in the mesoderm. Notochord is a part of the mesoderm, but its development is special. And new formation of red endoderm. It is not fully developed here, but we will see, no, it is much older embryo, but we will see fully developed endoderm here, and notochord in the center of the body. The same is here. Above is a developing neural tube, and also here it's developing neural tube, and directly around the notochord there is a formation of vertebral column. So vertebrae are developing here. Okay, the shape of the whole disc is changing during the third week more and more. It becomes still longer and longer, and not only flat. At the beginning it's quite flat, but now at the end, at the second half of the third week, some structures are protruding to the amniotic cavity, and the main of them are neural folds. Neural folds are structures which are made of ectoderm and which are highly protruding. I will show one picture where it will be visible from side, where neural folds are protruding. So the embryo is changing slowly from the flat disc, trilaminar germ disc, into the tubular embryo. Tubular shape of the embryo will be then typical for the fourth week of the development. Here is the picture of the embryonic disc, probably 18th day of the development. And neurulation is a thing which happens during the second half of the third week. Again, there are some reasons why for the formation of neural tube it's important low level of BMP4, which are inhibited or which is inhibited by these factors. Here you already know some of them from the primitive node, so primitive node plays a big role here again. Ectoderm in the central area proliferates, invaginates. Neural plate is here forming. This here is nothing at 15th day, but 18th day there is a neural plate in the central line. Neural plate becomes bigger and bigger, and groove is forming in its center, and folds are forming in the periphery. So at the end of the third week, it is the 21st day, we have the tube is forming. During the formation of tube, there is one interesting cell population which is developing from the rim of the tube, or let's say from the tips of neural folds. This is this orange population of cells which lose their epithelial organization and leave the tube and are then situated in the mesoderm, and these cells develop neural crest. We will use neural crest cells for the formation of many tissues and many structures, so please remember neural crest at this moment, and we will come back to them later. Neural folds, and they are very high, mainly in the cranial area where brain will develop. This picture was already here, but here, this is the picture which I like the best, where our neural folds very well visible, and you can see it is the end of the third week, and you can see that the embryo is now really tubular, not flat, not like a disc. So we are at the end of the third week, and our mesoderm, our intra-embryonic mesoderm, which was developed during the third week, has to develop more, has to differentiate. So the next chapter is differentiation of the intra-embryonic mesoderm. How does it differentiate? We know that the cells of intra-embryonic mesoderm are migrating from the primitive streak. Primitive streak is in the caudal area, and we can say that it has several segments. It has its cranial segment, it has its middle segment, it has its caudal segment, and it has also its terminal segment near the cloacal membrane. Cranial part of the primitive streak gives rise to population of cells which are migrating along the notochord. That means along the axis, and that is why this part of the intra-embryonic mesoderm is then called paraxial mesoderm. This PM means paraxial mesoderm. Paraxial mesoderm isn't simple one population. It is possible to divide it into more cranial, so-called head mesoderm, and less cranial somitic mesoderm. So paraxial mesoderm has got two parts, head mesoderm and somitic. Not somatic, but somitic. It is important difference. Somitic and head mesoderm. middle part of the streak gives rise to IM, which means intermediate mesoderm, which migrates also cranially, but not so much. This paraxial mesoderm can reach the oropharyngeal membrane. The migration of intermediate mesoderm is less extensive, so it migrating only to the area of cervical, to the cervical area. Then we have this green part of the mesoderm developed from this caudal end of the, not very end, but almost end of the primitive streak. It's abbreviated LPM, which means lateral plate mesoderm, or you can also use simply a lateral mesoderm. Lateral or lateral plate mesoderm. It is migrating very, very much, and it is migrating around the oropharyngeal membrane, and it is filling from both sides the area in front of the oropharyngeal membrane, which we already know as the cardiogenic zone. So the lateral plate mesoderm participates in the formation of cardiogenic zone. That means participate in the formation of heart and cavity around the heart, which is pericardial cavity. And then we have this orange end of the primitive streak, which gives rise to small population of cells forming a part of extraembryonic mesoderm. You already know extraembryonic mesoderm. It was developed during the second week from hypoblast. And now these cells, this epiblastic, in fact, epiblast is the origin of all intraembryonic tissues. So these cells now participate in the formation of new cells of extraembryonic mesoderm, because it has to increase in size and the number of cells has to increase. So primitive streak is participating in it. So there are several population of mesodermal cells, and this picture is called something like a fate map of the mesoderm. We know where where the cells from each part of the primitive streak are migrating, and where they are then staying, and where they are developing some structure. Another picture, another fate map from another textbook, which starts earlier. This picture down is analog to our previous picture. You can again see head, somitic, intermediate, lateral plate mesoderm. Here upstairs are two pictures showing the situation a little bit earlier at the beginning of the gastrulation. The first day of gastrulation, this was formation of endoderm, and then there is probably some not very clearly described cells of cardiogenic mesoderm, which are probably migrating to the cardiogenic zone earlier, before the stage of lateral plate mesoderm. So there are two populations of mesodermal cells which are participating in the formation of heart, which some of them are from so-called cardiogenic mesoderm, which is developing earlier, and some of them are from lateral plate, which is developing later. Let's look at each of these types of mesoderm in little bigger detail, but today we will mostly deal with paraxial mesoderm, and at the end of lecture with lateral plate. Intermediate mesoderm, there will be just one picture about it, and we will speak more about it in the formation of urinary and genital systems. Paraxial mesoderm is the thickest part of the mesoderm. The mesoderm in this central paraxial area is very thick, and the mesoderm here is forming segments. So there is the full segmentation, or almost full segmentation of paraxial mesoderm. You already know something about the segmentation from the lecture about ontogenesis. So insects are fully segmented, but humans are not so much segmented at the end of the development, but during development there are segments which are developing by the same methods as the segments or parasegments in Drosophila. These segments of the paraxial mesoderm are called somites, and that's why main part of the paraxial mesoderm was called somitic. You hopefully remember this. Somites are thick segments, big segments of the paraxial mesoderm. Intermediate mesoderm doesn't develop so quickly, so it is just small part, and lateral plate, it is developing very early, and it is splitting. Lateral plate is splitting by fusion of these intercellular cavities, and this new space is here developing. I will speak about it in a moment. At first, something about the paraxial mesoderm. Why is it segmented? It is segmented because there exists so-called segmentation clock. Segmentation clock is a molecular mechanism where mainly notch gene is important. You already know notch as a surface receptor, important for lateral inhibition, but notch is also important as a cyclic gene. Its highest level is developing in time, and when there is a highest level of notch, or the protein expressed from the gene notch, then a signal for formation of new somite is here. So somites are developing in very regular order. The first somites are developing during the 20th day, and then every next day 3 somites are developing. So the clock is very perfect, it's working very regularly, and so we can exactly recognize the age of the embryo between the end of the 3rd week and the end of the 5th week. So during these 2 weeks we can clearly recognize the age of the embryo about the number of somites, because every day 3 somites are forming. 3 pairs of somites, because somites are in both sides of the embryo, in both sides of the notochord. Big role is here played also by the gradient of fibroblast growth factor 8 and retinoic acid, again molecules which you already know. FGF8 is decreasing caudally, and retinoic, its level is increasing in the caudal direction, and the level of retinoic acid is the highest in the cranial end of every somite, or also in the cranial end of the embryo. The head mesoderm, which was one of the part of paraxial mesoderm, you hopefully remember it, doesn't segment so much completely, and so there exist 7 not fully developed somites which are called somitomers. So 7 somitomers are developing in the cranial area, and the first fully developed somite is the first occipital somite. There are 4 occipital somites. Oh, no, it isn't here written, but it doesn't matter. There are 4 occipital somites. There are then 8 cervical somites, 12 thoracic somites, 5 lumbar, 5 sacral somites, and not so exact number of coccygeal somites, which means that the total number of somites isn't stable. It's between 42 to 44. 42 to 44 somites are developing, and the first of them is the first occipital one. So not the most cranial end of the paraxial mesoderm. Here is a nice photograph from the scanning electron microscope where several fully developed somites are visible. They are along the neural tube, and under the neural tube is surely not a chord, but we don't see it. And here you can see that this somite is just forming. It isn't fully developed, it isn't fully separated from the neighboring somite, because it is just happening, this separation. Here is a high level of the FGF, here is a high level of retinoic acid, and that's why the border is forming between these two places. I already said how and when, in which rate the somites are developing, so that was said. And now we go on. Somite at the beginning is a structure composed of epithelial cells. Why epithelial? Because when the original mesoderm wasn't epithelial, there were migratory cells, but these migratory cells are joining together and forming epithelial tissue after the finishing of the migration. These epithelial cells are forming spherical groups, which are called somites. These groups at the beginning are hollow. There is a cavity in the center of the somite, which is called somitocell. Sometimes some special cells are found in the cavity. Sometimes this cavity is describing or is described as free. It's not so clear. And the whole somite then resembles something like a donut and has a laterodorsal wall and ventromedial wall. Laterodorsal wall is preserved longer. Its epithelial structure is longer preserved, but ventromedial wall becomes mesenchymal tissue very early. You already know mesenchyme. What is it? Mesenchyme. Mesenchyme is a primitive connective tissue, embryonic connective tissue. This tissue can develop from many sources, and one of them is this ventromedial part of each somite. This part of the somite, which is very early transforming into the mesenchyme, is called sclerotome. Here is the sclerotome, and it will be later a source of bone and cartilage mostly. It is mesenchyme here. The lateral part of the somite can be called dermomyotome. Dermomyotome later differentiates into two parts, myotome and dermatome. Dermatome also transforms into the mesenchyme and gives rise to connective tissues of skin and also subcutaneous layer. Myotome doesn't transform into mesenchyme. Its cells become myoblasts, and it is the source of skeletal muscle tissue. And all skeletal muscle tissues of the body are derived from myotome. So it is a thing which you should remember, that there doesn't exist another source of skeletal muscle than paraxial mesoderm, and particularly its myotome. Somites in the pictures. At the very beginning, this is just thickening of epithelial mesoderm. Later, lumen is here forming somitocell, so it's like a donut now, again, donuts. And here you can see that dorsolateral side is still epithelial, but here epithelium disappears and mesenchyme is here forming, so dispersed cells and some extracellular matrix. So here is sclerotome and here is dermomyotome, which also loses its epithelial arrangement but later, and gives rise to dermatome and myotome. This neural tube in the center. Notochord is here, which becomes a part of the future vertebral column. Here is endoderm, here is a surface ectoderm, yeah, and a somite on the other side. So somites are symmetrically situated in both sides. Some molecules again. The main molecule important for the transformation of epithelium into the mesenchymal tissue in the area of sclerotome is your well-known sonic hedgehog, expressed and secreted from notochord and so-called floor plate, or yes, floor plate of the neural tube. Main factor important for the transformation of myotome into myoblasts is VNT, again well-known signal molecule. Wasn't anything in the previous lectures about NT3 neurotropin 3. It is a factor which is important for the stimulation of PAX3 transcription factor, important for the formation of dermatome. Okay, it isn't so much important. I think that sonic hedgehog and VNT should be the most important for your study in this picture. One picture of the intermediate mesoderm. Intermediate mesoderm develops a little bit later, but then it also segments. It becomes segmented, but not along the whole length, but only in the area of cervix, future cervix, where segments of intermediate mesoderm are called nephrotomes. The other, that means thoracic, lumbar and sacral part of the intermediate mesoderm remains unsegmented, and it's called nephrogenic blastem. Nephrogenic blastem, which gives rise to kidneys and gonads and some parts of genital ducts also. So it's everything at this moment about the intermediate mesoderm. We will speak about it in urogenital system. And the lateral plate mesoderm, it will be more discussed at the end of the lecture, but now at this moment it's probably enough to say that this plate is completely unsegmented. There is no segmentation in the lateral area of the mesoderm, but instead of segmentation there is formation of intercellular cavities. Cavities are forming in the mesoderm, which fuse together to form one big common space, which isn't only common in the lateral plate, but it's also joining the extraembryonic coelom, which is here, it is here written, extraembryonic coelom, which is also known under the name chorionic cavity, you already know it. And this new space here is then called intraembryonic coelom. So a new space is forming between two layers of lateral plate mesoderm, and these two layers here have the same names as the layers in the wall of extraembryonic coelom. So again, somatic and splanchnic, or somatopleuric and splanchnopluric mesoderm. Somatopleuric or somatic mesoderm is connected with the ectoderm, and splanchnic or splanchnopluric mesoderm is joined to endoderm, and intraembryonic coelom is a cavity in between. But now at this moment it isn't full cavity because it is connected by this narrow junction with the extraembryonic coelom. Okay, so we can stop the description of mesoderm itself and let's focus on the formation of some systems which are developing from the mesoderm. Let's start from skeleton. Most skeleton is from mesoderm, and the main source, the main mesodermic source of the mesenchyme giving rise to bones is a scleroton. You already know it. That means ventromedial part of a somite. It isn't important here to know the parts of the scleroton, ventral, lateral, central, dorsal. No, it isn't important. This picture is here just for illustration that the tissue of the scleroton, which was before epithelium, but now it is mesenchyme. That means loosely organized migratory cells and some extracellular matrix. These cells are migrating to all spaces around the neural tube, even between the neural tube and ectoderm, and also around the notochord. And so all the area around the neural tube and notochord is filled with mesenchyme of this origin. This mesenchyme can then transform into bone or into cartilage. It would be quite simple if we say that each pair of somites gives rise to one vertebra. It would be fine, but it doesn't work so simply. Sclerotons have to resegment. Resegmentation of scleroton. It is very important for the formation of vertebral column. If scleroton doesn't resegment, vertebral column wouldn't be stable and we wouldn't move, never. So, what does it mean, resegmentation of scleroton? Each scleroton, that means each ventromedial part of each somite, divides into two parts. One would say two halves, but one of these parts is smaller, the cranial one, the caudal one is bigger, so it isn't exact division into two halves, two parts, cranial and caudal. Each caudal part fuses with neighboring cranial part. You can follow these arrows. So there is fusion in this area. And so these two things together establish a body of the vertebra. Of course, there must be the same structures at the other side, of the neural tube and notochord. So the vertebra is formed from why does it happen? Vertebra is formed from four structures: two cranial parts of sclerotomes and two caudal parts of sclerotomes. Why formation of vertebrae is so complicated? This is the derivative of myotome, future short intervertebral muscle. If each vertebra is developing from one somite, one sclerotome, this muscle would be connected with the cranial and caudal end of the vertebra. It wouldn't bridge the intervertebral space, and so it wouldn't work. It wouldn't hold the vertebral column, and the vertebral column would disappear. The same would be with ligaments between vertebral columns and vertebrae and so on. But as the resegmentation exists, the muscles are then bridging intervertebral spaces. On the other hand, nerves from the spinal cord can go through these intervertebral spaces. And when we look, this is from another textbook, but again, fusion of caudal and cranial part of the sclerotome, forming one half of vertebra. Intervertebral nerves are going through the intervertebral spaces, and then there is a fusion. Intervertebral discs are forming in the areas where before the central parts of sclerotomes were. Nucleus pulposus is then developed as a remnant of notochord, and when we look, it was in one of these pictures, or no, it doesn't matter. When we look to formation of arteries, which were originally intersegmental, they will go to the vertebral bodies and not to cartilaginous intervertebral disc, which is cartilage is avascular, so there no arteries are necessary, but bone is well vascularized, so you need these arteries in the vertebral bodies, so everything has a sense. Vertebrae are then ossifying at first. Cartilaginous models are here forming several ossification centers, primary, secondary, and the special shape of each vertebra is formed. So vertebrae are made from sclerotomes, and each vertebra is made of four parts of four neighboring sclerotomes. Two left, two right. And here is the photograph, special staining. Blue is cartilage, so blue are intervertebral discs with nucleus pulposus in the center, and red is here developing bone tissue. So this is the body of the future body of the vertebra, developed from two sources, cranial and caudal. Ribs are derivatives of vertebrae, so they also originate in sclerotome, so in Paraxial Mesoderm. On the other hand, sternum, which is connecting ventral sides of ribs, is developed from several ossification centers developing in the lateral plate mesoderm, particularly in its somatic layer. Somatic or somatopluric layer of the lateral plate is the origin of sternum. So sternum and ribs are of different origin. Skull skull has a very interesting origin, and so we have to say at first something about the classification of bones of skull. Bones of skull can be classified very simply according to anatomy. You already know it. They are classified as the bones of neurocranium surrounding the brain, and splanchno or viscerocranium, I don't know what exactly do you use in anatomy, which is around the cranial end of digestive tube. So it is anatomical classification of the cranial bones. The second is developmental classification of cranial bones, or histogenical classification. That means bones which are originating by intramembranous ossification, they form, or they belong to so-called desmocranium, and then there are bones which are endochondral, and they are forming chondrocranium. Some bones are completely intramembranous, like parietal bone. Some are combined, like for example temporal bone, which has chondrogenic basis and membranous squama. It's possible to say that most bones of the calva are intramembranous, so desmocranial. Most bones of the viscerocranium are desmocranial, and endochondral ossification is found mostly in the base of skull. Temporalis, sphenoidalis, ethmoidalis. These are typical endochondral bones. And also endochondral bones are middle ear ossicles, incus stapes malleus, and hyoid bone. And here we can combine these two criteria, what belongs to neurocranium, viscerocranium, and desmocranium or chondrocranium. Each color belongs to another type of bone, so you can see the same what I already said. And photograph, where blue color has a cartilage. It is very early stage, so this is the Meckel's cartilage situated at the place of future mandible, which doesn't give rise to mandible directly. So mandible is developing around the cartilage, but is mostly intramembranous. It belongs to desmocranium, and here is the base of skull, future occipital, temporal, sphenoid, and ethmoid bone. Here is developing capsula otica. Ear is here forming, which belongs, capsula otica then gives rise to Petcher's bone, which belongs then to temporal bone. If we look to cartilaginous neurocranium, that means to base of skull, we can see several cartilages which can be important: parachordal and prejordal cartilages, and some so-called capsules. Capsules are developing around the sensory organs, so there exists olfactory capsule, optic capsule, and otic capsule around the future inner ear, and they transform into some parts of skull: ethmoid bone, sphenoid bone, temporal bone. For embryology, is probably the most important, the last classification of the bones of skull, according to origin, according to the source of their mesenchyme. Some of bones are developing from paraxial mesoderm and mostly from somitomers, which are developing at the cranial end of paraxial mesoderm in the so-called head mesoderm, and also occipital Somites are participating in the formation of skull. So, paraxial mesoderm. The second source of mesenchyme for the skull is already mentioned neural crest. I really briefly mentioned that this is the population of cell which is forming during neurulation from the rims of neural groove, so that means from neural folds. These cells here lose their epithelial organization, leave the neural tube during its closure, and establish cords of loosely organized cells around the neural tube, which is called neural crest. Neural crest. Neural crest can develop in many ways. It is visible here. Neural crest can develop towards glial cells, for example, Schwann cells or satellite cells in ganglia. Neural crest can develop into melanocytes which migrate to skin. Neural crest of trunk can differentiate into adrenal medulla, for example, or neurons of dorsal and vegetative autonomic ganglia. And very important is neural crest in the area of head. It also gives rise to ganglia or meninges around the neural tube, but also to mesenchyme. And this mesenchyme is important for formation of some bones, connective tissues of eye, connective tissues of so-called pharyngeal arches in the area of neck, odontoblasts, which are the cells of the tooth, and also one structure which is quite distant from the head, and it is septum in the heart. A part of the interventricular septum has the same origin because the cells can migrate in a very long distance. So many structures are developing from neural crest of head, and these which are developing through the mesenchyme are developing from so-called ectomesenchyme. It is a term which is used for the mesenchyme of the ectodermal origin. Ectodermal origin of mesenchyme is only through the neural crest. So neural crest is the source of ectomesenchyme. And some bones of skull have got this origin. So some bones of skull are from ectomesenchyme, derived from neural crest, and another bones are from paraxial mesoderm. When we look at the basis of the skull, we can see a clear border between these blue parts, which are from neural crest, and the red bones, which are from paraxial mesoderm. This border is Pituitary Fossa, or sella turcica in the sphenoid bone, where pituitary gland is situated. So at this level, where is also the end of former notochord. Notochord was finished here because pituitary fossa is at the place of former oropharyngeal membrane. Paraxial mesoderm, which was under the influence of notochord, formed bones situated caudally from the pituitary fossa, and neural crest gives rise to bones cranially situated. If we look at the whole skull, we can see the border between frontal and parietal bone. Parietal bone is from the somite, or somitomer. Frontal bone is from neural crest. Also here it's a little complicated in the temporal bone, which squame is from neural crest, but petrous part is and base is from paraxial mesoderm. Okay, so it's very important and very interesting. What can be the source of bones of skull? Picture of a bone showing or picture of a skull showing formation of the parietal or frontal bone by intramembranous ossification. Now let's look at the muscle system. Muscle system, I mean skeletal muscle system only. At this moment I will speak only about skeletal muscle. So all skeletal muscles are developing from the paraxial mesoderm. It's fine that there isn't any other source, fine for students. And the particular part of the paraxial mesoderm giving rise to skeletal muscles is the myotome. Myotome, which is developing from two anlages, two origins in the dorsolateral part of a somite, which are under the influence of VNT, which can be secreted by neural tube and which can also be secreted by lateral mesoderm. These two parts of the myotome then fuse together. You can see it on the next picture. Nevertheless, they still differ. There are still another signal molecules expressing, and one of them, this more medial, gives rise to so-called epaxial muscles, and the second is lateral-ventral part of myotome gives rise to hypaxial muscles. So muscles can be epaxial and hypaxial because of the formation from two parts of a myotome. There exists also another classification of muscles, primaxial and evaxial. It's more complicated. Primaxial muscles are developing and also originating within somatic mesenchyme, but abaxial muscles, they arise within somatic mesenchyme because there doesn't exist any other possibility, but migrate, their myoblasts migrate to somatic lateral mesoderm, and these muscles are developing under influence of the somatopleuric mesoderm, of somatic mesoderm, and so these muscles are a little bit different. These abaxial muscles have to cross this line here, which is called lateral somatic frontier. So muscles which are developing from myoblasts migrating through this frontier will be abaxial, and these which don't migrate through this area or through this line or border frontier will be primaxial muscles. So it means that all epaxial muscles are primaxial, but only some hypaxial muscles are primaxial and other ones are abaxial. Epimeric or epaxial muscles give rise to extensors of spine, long and short extensors. Nothing more. So all other muscles are hypaxial. In the area of trunk, they give rise to 3 layers of intervertebral or abdominal muscles, also musculus rectus abdominis is hypaxial, and They migrate, or their myoblasts migrate also to limbs. So extensors and flexor groups of limbs, all the muscles of limbs are also hypaxial. And of course apaxial, because they are developing in the lateral mesoderm, giving rise to the tissue of the limb part. Little bit different are cranial muscles. Not all. Some of them are developing from occipital myotomes, so the situation is normal here. Mostly muscles of tongue are developing of occipital myotomes, but another muscles in head are developing from unsegmented or only poorly segmented somitomers. You know that there are 7 somitomers cranially, and cranial muscles, for example, oculomotoric muscles, muscles of pharyngeal arches, that means masticatory muscles, mimic muscles, muscles of middle ear, muscles of pharynx. All these are from myotomes in the unsegmented paraxial mesoderm, forming 7 somitomers. Myoblasts are migratory cells. That is why muscles can be everywhere, can migrate from paraxial mesoderm to tips of limbs, for example. When myoblasts finish their migration, they fuse. So the second step in the muscle fiber formation is myoblast fusion. Myoblasts are fusing in one syncytium. You know that muscle fiber is syncytium, but at first this syncytium has got central nuclei. It is the cross section. You can see central nucleus, and this for future muscle fiber which has still central nuclei is called myotube. So one developmental step of the formation of muscle fiber is myotube. Central nuclei, myofibrils are slowly developing, and when myofibrils are filling the central part of the myotube, nuclei are transported to the surface. So in the final stage, the mature muscle fiber has got peripheral nuclei. So it is the difference of myotube, which you can find in embryos or fetus, and the muscle fiber, which you can find after birth and in adult. Here are myoblasts. You can see single cells. Each has got one nucleus, and this picture isn't very nice, but the authors of this picture say that these are myotubes. Okay, maybe, but I don't see exactly. Maybe here I see a nucleus, and it is really in the center, but okay, let's skip it. So muscles were developed. Fine. Let's go to development of limbs. Limbs are structures which are developing little later than the others. They start to form at the end of the 4th week. This is the embryo at the 25th day. It still hasn't got any signs of limbs, but the embryo at the end of the 4th week, 28th day, has got one limb ridge. Of course, it has 2 limb ridges. Another one is on the other side. These 2 limb ridges are developing at the level of the 5th. Is it 1, 2, 3, 4 somites? So 5th somite, 6th somite, 7th, 8th. Let's say 9. So at this level of the cervical somites, one limb ridge is developing, and it is of course the limb ridge of future upper extremity, cranial extremity. Some textbooks say that it is 26th day, 27th day, so let's say at the end of the 4th week. About 2 days later, another ridge is forming at the level of lumbar and sacral segments, and it is a ridge for the lower extremity. The position of the ridge is given by Hox genes, by their expression, which is different along the craniocaudal axis. So upper limb earlier, lower limb later, about 2 days later, everything at the end of 4th week or maybe very beginning of the 5th week. And here we can see limb. It was limb ridge before, but limb ridge is quickly transforming into limb buds. And here is a nice picture of the embryo at the beginning or during the 5th week, and it has a limb bud, or here is the histological section of limb bud. And in this high magnification of limb bud histology, we can see thickening of the ectoderm on the surface. It is so-called apical ectodermal ridge, AER. Apical ectodermal ridge is a thickening of the ectoderm, and it is the inducing center of the whole ridge, or later bud, because this epithelium here secretes molecules, mostly fibroblast growth factors, which are important for proliferation and later differentiation of mesenchyme here. The origin of mesenchyme in limb ridge or bud is lateral plate, and of course it's somitic layer, somitic, which is connected with the ectoderm, so somitic layer of lateral plate. During the 6th week we can see something like a constriction in the middle of the limb ridge. Of course, again, at first in the upper extremity. So upper extremity at this case has 2 segments. Lower extremity still hasn't got any constriction because it has still delay. It has still 2 days delay compared to upper extremity. So there are 2 segments, and these segments are called autopodium. Sorry, the autopodium is here. So the first, this proximal is axopodium, or maybe can be simpler axopod, and distal autopod or autopodium. Autopod and axopod are parts of 2-segment limb, and autopod is later transforming into the palmar or plantar plate. Axopod is developing to the remaining parts of the limb. So it must transform into new segments during the next week. Here is the 7th week, and you can see that former axopod has got this angle here. It is the area where elbow or knee is. It depends on the extremity. And now this proximal part of the axopod will be called stylopod or stylopodium, and this middle part is now zeugopod. And here is still autopod, already transformed into palmar or plantar plate. I mean that in this case palmar plate because it has the shape of future hand. And the proof of cartilage, proof of chondroitin sulfate here, is showing digital rays of the autopod 5-digit arrays, future fingers, and also tubercles and grooves between them are here visible. These grooves become deeper and deeper, and it is the typical participation of programmed cell death, that means apoptosis, in the process of formation of hand. So formation of hand and formation of these interdigital grooves is a very good example of progressive effect of apoptosis in the development. Oh, sorry, opposite direction. Yes, and here are these terms written. So axopod and autopod, or axopodium and autopodium in the 2-segmentary limb, and new zygopod and stylopod in 3-segment limb, giving rise to femoric or femur or humerus, and of course zygopod gives rise to forearm or this part of lower extremity. Okay, skeleton, that means bones, cartilages, joints of the extremity is from the somatopleura, somatic lateral mesoderm, but the extremities have got another tissues also. There are nerves, there is skin, there are muscles. So some other sources, some other origins of tissue play a role in the formation of extremities. Muscles are of course from Somite, from Paraxial Mesoderm. Nothing else is possible. Motor nerves are axons of neurons from neural tube, but sensitive nerves are axons of neurons from neural crest, because they go from the sensitive ganglia. Vessels are growing into the limb buds. Pigment cells, melanocytes are migrating from neural crest. So there are several other sources, but main source of tissues in the limb is ectoderm on the surface and lateral somatic mesoderm centrally. It is quite well described, the molecular direction of the molecular regulation of the limb development. So we know that the first factor staying at the beginning of the limb formation is FGF10. Then the crucial role is played by factors secreted by ectodermal source of apical ectodermal ridge, mainly FGF4 and 8, and the differentiation of the craniocaudal axis of the limb, that means where will be thumb, where will be small finger, is directed by a special group of cells which are at the caudal end of the bud, and this is called zone of zone E is here, of polarizing activity, ZPA, and it is well known about this zone that the cells here secrete sonic hedgehog. Sonic hedgehog is secreted by cells of mesenchymal cells, of course, of ZPA, and the high level of sonic hedgehog is a signal for the tissues here to differentiate towards the tissues of the caudal fingers and ulnar side of the forearm, for example. And low level of sonic hedgehog gives rise to formation of thumb and radial side of the forearm. So it is quite interesting. Also, dorsoventral axis is established at the very beginning. So everything is quite well understood here. Apoptosis, that I already said, that is important for the formation of fingers. And limbs will rotate. They will rotate during the fetal development. You can see 10th week, that means young fetus. Upper limbs will rotate by 90 degrees laterally, which means thumbs will be then at the lateral side in the basic anatomical position, and on the other hand lower limbs, feet, will rotate in the opposite direction 90 degrees medially, and so toe is situated at the medial side of the foot in the basic. No, it is not dependent on the position. It's every time on the medial side of the foot. Okay, of course. Okay, so let's go. I know that it's maybe a little longer, but let's go to last part of my lecture, which is development of coelom and diaphragm. Coelom means intra-embryonic cavity. Intra-embryonic cavity, which you already know. Where does it develop? It is developed from intercellular small clefts, small cavities found in the lateral plate mesoderm. Lateral plate mesoderm is unsegmented, but intercellular cavities are here formed. They slowly fuse. No, not slowly. Slowly is a bad word in embryology, because you can see this is 19th day, this 20th day, so 1 day older, and here are single cavities, and here is everything fused into the common space. So yes, I must repair myself. It is very fast fusion of these clefts or cavities, and space is developed, which is at this time widely connected with the extraembryonic coelom. So there's this connection, this junction. This junction is found laterally, but our coelom is developing also cranially here in cardiogenic area, because lateral mesoderm was migrating during gastrulation here as well. But here in this cranial area there isn't junction of the coelom and extraembryonic coelom, of the intraembryonic and extraembryonic coelom, because there is one additional layer. I will show it in a moment. So the intraembryonic and extraembryonic coelom is fusing only, or are fusing only laterally in both sides. And you can see that after the formation, the coelom has got a shape of a letter U or horseshoe. Horseshoe is typical shape of the coelom, and it's open laterally here and here, but closed here cranially. Here it is separated from the extraembryonic space. This coelom, intraembryonic coelom, will develop during the embryonic period into several other cavities. It will give rise to pericardial cavity, it will give rise to peritoneal cavity, and these pericardial and peritoneal cavity will be connected by pericardio-peritoneal canals in both sides. There are two canals which transform later into pleural cavities. Pleural cavities. How can it happen? It happens because there is the embryonic folding. embryonic folding is a very important process of the 4th week. Embryo is already tubular. Its tubular shape was given at the end of the 3rd week, and then it folds because it grows faster than is the surrounding space allowing, and so it cranial, caudal, and lateral poles are going down, going ventrally. Everything is folding to the ventral side. There are then 2 lateral body wall folds, and the next picture will show cranial and caudal wall folds, and all structures which are originally cranially and laterally become ventrally situated and become concentrated around the yolk sac, which is still here and which becomes narrower. We will speak about it more when we will speak about the formation of digestive tube and other structures. So now let's stay at this point that there is lateral and craniocaudal folding, and the coelom, which is laterally widely connected to the extraembryonic coelom. Here is, let's say, a border of the intra and extraembryonic coelom, and you can see this wide communication here. So these 2 parts of the coelom become separated by this folding. This communication is here only small, and here no communication is because these folds were fusing. Of course they were not fusing everywhere, but usually fusing, and so finally we have got real intraembryonic cavity, and not only let's say half cavity open to the surrounding space. Now we can look to the same folding in the craniocaudal direction. So here is cranial fold, caudal fold, and they become situated closer and closer. And I add one structure to this schema from your textbook, which is called septum transversum. I didn't add this structure, it was already here, only this label wasn't here. Septum transversum is a plate made of lateral mesoderm, which is situated between the intraembryonic coelom and extraembryonic coelom in the cranial area, in the cardiogenic zone. This septum transversum here closes the communication between intra and extraembryonic coelom from the very beginning. And it goes ventrally together with other structures in this cranial side. So septum transversum is here the most cranial thing of the embryo. Here it slowly goes more ventrally, and this goes still more and more ventrally. It becomes, here it is situated in the area of neck. Here it is then situated between thoracic and abdominal area, and it is still the same septum transversum. It's just changing its position, and of course grows during these, let's say, 10 days, but it is still the same tissue. Septum transversum is very important for the separation of thoracic and abdominal cavity, because it gives rise to biggest part of the diaphragm, and it is also important for the formation of liver. Liver is growing into the liver bud is growing into the septum transversum, and so it is important for the formation of this organ as well. So after folding, that means at the end of the 4th week, we have got the intraembryonic cellum separated from the extraembryonic cellum, but it is still one tubular space, one horseshoe-shaped and curved tubular space. And the last part of my lecture, really two or three last pictures, will describe how this single tubular space is then divided into definitive cavities. Let's start by separation of pericardial cavity from pericardio-peritoneal canals. Pericardial cavity, which is surrounding heart, which was originally cranially, but after folding in the 5th week, it is situated ventrally, is separated from pericardio-peritoneal canals by special folds, which are called pleuropericardial folds. They are developing from the lateral wall of the body, that means from somatopleura. They are developing by the growth of lungs. Lungs are growing into the pericardio-peritoneal canals, and they press the tissue laterally to form this evagination, or invagination is better to say. So it is one motor of formation of pleuropericardial folds. Another motor is the existence of special common cardinal veins. We will speak about the development of veins later, so it doesn't matter that you don't know what is it, but common cardinal veins are situated in the pleuropericardial folds, and they tend to go to the central area and fuse. So it is the second motor, and the result is formation of pleuropericardial membrane. So these two folds fuse here. The next picture, it is almost fused, and here it's completely fused. Also veins are fused, giving rise to superior vena cava. It's here, superior vena cava. Oh no, it's nonsense. It must be aorta. Superior vena cava is really here. And because here is vertebral column. And so the heart is then in completely closed cavity, which is called pericardial cavity. So pericardial cavity is now separated from remaining parts of the coelom. During this time, lungs were growing. Lungs are developing little later than other organs because embryo or fetus doesn't need them. It doesn't breathe, and so they can develop later and longer. And so we will speak about the development of lungs, so don't worry. You will hear how does it happen. And lungs, when growing, are increasing the pericardio-peritoneal canals, and they are transformed to pleural cavities. So here you can see the name pleural cavity, which was originally pericardio-peritoneal canal when narrow. And we will go a little bit more caudally to the caudal ends of pericardio-peritoneal canals, and let's separate the peritoneal cavity from pericardio-peritoneal canals. It is the area where diaphragm is developing. Diaphragm is developing from similar sources which were in the previous separation. Pleuroperitoneal folds. Everything starts by P, so it's very confusing. Previous folds were pleuropericardial. These folds here are pleuroperitoneal. They are also derivatives of the somatopleura. Lateral body wall is invaginating here and fusing, but in this case they don't fuse together, but because there is a mesentery between with esophagus and vessels, aorta and inferior vena cava in this case, and so these mesentery and the tissue around esophagus and these big vessels become the second part of the future diaphragm, and the third and probably the main part of future diaphragm is this orange part, giving later rise to centrum tendineum of the diaphragm, and it is septum transversum, which was explained, or the origin of which was explained before a while, which originates in the most cranial end of the embryonic disc and then is moving together with other structures downstairs and caudally during the embryonic folding. And septum transversum originates in splanchnopleura. And the last, let's say fourth, source of tissues found in the diaphragm are muscles from the body wall, and because the diaphragm is developing or starts it developing when present in the cervical area, these muscles here migrate from cervical somites, and that's why diaphragm situated in fact at the border of thoracic and lumbar area is innervated from phrenic nerve or biphrenic nerve coming from segments C3 to 5. So that's the origin of diaphragm. And I think it is the last picture. Yes, really, it is the last picture of my lecture. So thank you for your attendance. I hope that you were able to understand my lecture, but don't worry if something was too fast. There will be a practical training about it where we'll try to explain everything. We'll use some video movies where maybe these dynamic changes like this embryonic folding will be explained better than by static pictures of my lecture. So don't worry. I hope you will understand everything. Read the textbook and come prepared to the practical training, or in your case it will be distant online practical training by Zoom. Nevertheless, read textbook and be prepared, be ready to try to understand it, because it's quite complicated topic, this second general embryology. There will be another lecture, embryology 3, so we will try to explain the things from both lectures in this practical training. So, goodbye.